Uh, Today, we are going to discuss about overview of genetic engineering and its impact on society. This is particularly for human society, and now, like these are the major concepts I am going to cover today. The concept of genetic engineering, and then followed by genetically engineered products for human uses, and a brief overview of tools and techniques required for genetic engineering. So in this module, module 11, so if you can remember the last two lectures, mostly I was discussing about some techniques which are very important for genetic engineering. In lecture 51, I was discussing about agarose gel electrophoresis, where you can analyze different types of DNA molecule based on their size, and also polymerase chain reaction, where you can amplify a particular DNA sample. And in lecture 52, we are discussing about DNA sequencing, like whenever you are doing some kind of genetic engineering or you are kind of trying to manipulate some genes, then after manipulation, always we have to check whether our manipulation is right or wrong. So that's why DNA sequencing is also very important. So that's why I cover those three topics first before the actual discussion about genetic engineering. So what is genetic engineering? So if you see, genetic engineering means manipulation of an organism's genome, and this manipulation can be of different types. For example, it can range from changing one base pair. For example, I would say if this is our DNA, and you know that we have two strands in our DNA, right? This is 5 prime and 3 prime, 3 prime, 5 prime. So if I write some sequence like this, so some sequence there. So I would say this is part of a gene. The gene might be much longer than this, but this is just a segment of a gene. Now in this gene, what I'm trying to say here, that we can just change one base pair. That means if I concentrate here, for example, we have TA, so we can change it to, like in this strand, on the top strand we can make it A, so the lower strand will be T, or on the top strand we can put G and the lower strand should be C based on A, T and GC base pairing. So this is just one base pair change. Or what we can do is deleting a whole region of DNA. So for example, in E. coli, we have just one DNA molecule, one circular DNA, and I would like to make some kind of E. coli where I would like to delete a particular gene for my own interest. Like whatever research I am doing in my lab, I would like to delete one gene. That is also an example of genetic engineering. And not only deleting, the other thing is what we can do, we can introduce an additional copy of a gene. That is also very common. Sometimes to give, to attribute an additional property, we can provide some extra copy of a gene. So that is very common, and I will discuss in more details in today's class itself. And now, if we go a little bit more, we can also think that extracting DNA from an organism's genome and combining it with the DNA of another individual. In the previous point, I was telling about a same organism, but here, we can take some genetic information, some gene for a different organism, and you are putting into a different organism. That is also very usual and useful in genetic engineering. We do that very often. And here, all those things we are doing because we used to enhance or modify the characteristics of an individual organism. So that is our kind of goal for this genetic engineering approach. And now, first I would briefly discuss about the about genetic engineering at its impact on human well-being. So like genetic engineering, like we use a lot in agricultural application for long time, I would say. So the thing is, agricultural application, there are many examples, but I would give you some classic example of genetic engineering. So here, for example, like nowadays, whatever cotton we cultivate even in India, so those cottons are called BT cotton. BT cotton. So BT cotton is like this is now very available everywhere in US, China, in India, everywhere BT cotton like being used. So now if you see, this term came from this BT, means this is a name of a bacteria. So this is Bacillus thuringiensis. So this is a bacteria. Now B and T came from here. That's why this is BT cotton. So what happened? This bacteria, they produce, like this bacteria can produce some kind of toxins, like lot of toxin, different, almost like more than 100 different types of toxins available in this bacteria. So those are commonly called BT toxins, because Bacillus thuringiensis toxin. And many of those toxins are actually very toxic against insect. So what scientists, what researchers they have done, they have taken some of these toxin producing gene. So they have taken some of these toxin producing genes, okay, from this bacteria. So they have taken some of these genes and they integrated with the cotton plant. So as a result of that, this cotton plant, they are making this toxin everywhere in their cells and tissues. So as a result of that, when those cotton plants grow, those insects, particularly cotton bollworm, if they actually eat leaves of this cotton plant, they will not get mature, they will die actually. So this is very good pest control mechanism here in this case, I would say, through genetic engineering approach. So as a result of that, you don't need to spray insecticide or pesticide from outside. It is already inbuilt, it is already present inside the cotton plant itself. So this is one example. Genetically modified sweet corn, it produces huge amount of sugar and starch, and so there are a lot of different types of example available which we are nowadays using in agricultural field. Then the application in dairy and poultry farms, I will not take much time here. So for example, you know that different types of hormones are routinely used to increase the amount of, for example, amount of cow milk, right? And also to for the growth of poultry, all those things, like they are very much dependent on growth hormones. So I would say these are growth hormones, and these hormones are produced through genetic engineering approach. So we are making those hormones in a lab or in some pharmaceutical industry, and then we are actually, like, they are commercializing this growth hormone for this dairy and poultry farms. And now a third one, environmental application. There are many different types of application. I am just giving you some kind of overview, some important example, that's all. So environmental application. So naturally, some bacteria they can metabolize hydrocarbon, but scientists, they engineered those bacteria in such a way that those bacteria are very stable, and also they can, like, metabolize hydrocarbon very nicely. Like hydrocarbon means they can even metabolize petroleum. So as a result of that, these are generally called, commonly called oil eating bacteria. For example, this one. So if you see Pseudomonas putida. So this is one bacteria, and scientists engineer this bacteria in such a way that it is very stable, and also it can metabolize hydrocarbon petroleum very nicely. So what is the benefit? The thing is, this kind of bacteria like we can use in different types of, and like clearing environmental pollutants. For example, in oil spillage, there are these kind of bacteria can be used so that they can scavenge those oil, they can metabolize those oils. So as a result of that, it is I would say much better mechanism to take care of this oil spillage. So I would like to particularly mention that this oil eating bacteria and this Pseudomonas putida, this strain was actually like designed and mostly done by one of the Indian scientist. His name is Ananda Mohan Chakraborty. So he contributed a lot in making this oil eating bacteria. Okay, and therapeutic application. So now many therapeutic products are being made through genetic engineering approach, but one of the very important product, which is all of us we know that insulin. Insulin, particularly I would say human insulin. Human insulin was produced through genetic engineering approach in bacteria, and that insulin nowadays we are using to treat diabetic patient. So this is one of the very important example of therapeutic application. Apart from that, there are different types of growth hormones are also available for therapeutic application. So these are some examples that what you can do through genetic engineering approach. And again, some genetically modified product, some interesting product I mentioned here. For example, if you see this tomato, this is like called flavor saver tomato. So as you can see, this is genetically modified tomato, and the idea here is these tomato are modified in such a way that their outside wall, this part if you see, this is much thicker. Okay, this is thick, and during ripening process it will not be very thin soon. It will take some time. So the idea is, so they engineered some enzymes in such a way that this process will be slow. So as a result of that, this tomato should be very, I would say, like not be very soft. So the shipping is very easy, and for long storage, this is also very good, right? So this is genetically modified tomato. And this one, that zebrafish, as you can see, this is called zebrafish because of this kind of stripes, like you can see the stripes like zebra. And this zebrafish is very common in, I would say, in Southeast Asia. For example, in India, Pakistan, Bangladesh, this is very common. And what researchers they have done, this piece already they are, they look nice, but in addition to that, what scientists did, they put some additional genes from some other organism. For example, from jellyfish, right? Jellyfish are of different types of color. So they took some genes from jellyfish and then they incorporated in zebrafish. So as a result of that, from zebrafish now we made this glowfish. Okay, glowfish, they are glowing. So as you can see, people are keeping these glowfish in their aquarium nowadays. It's very much available commercially. So you can see here, for example, this one, this fish, right? This one. So it's green in color. So this zebrafish, this is expressing some protein. This is called green fluorescent protein, GFP, green fluorescent protein. Similarly, this one, this fish, this is expressing YFP, yellow fluorescent protein, and this one expressing RFP, red fluorescent protein. These proteins we use very frequently in lab for different purpose, but here we are kind of discussing in a different context that they change the color of this space and people are keeping this space in aquarium, that's all. Now, production of human insulin. This is the best example of genetic engineering, which is also called recombinant DNA technology. I would like to briefly summarize human insulin, how people are using human insulin in past and how we are making insulin in modern time, currently. So here the production of insulin in 1921, Frederick Banting and Charles Best, they successfully purified insulin from a dog's pancreas. And over the years, scientists made continual improvements in insulin production, because insulin, you know, that very important therapeutic reagent, because many of us, we suffer from diabetes. So insulin is absolutely required when the diabetes is really difficult to manageable by medicine. So now the thing is, later on many scientists, they also purified this insulin from pig, from horse, different types of animal. But the problem is you have to sacrifice a lot of animals. That is one thing. And the other thing is, although those are insulin of those animals, but still they are foreign protein. So whenever human, they are using this protein, this insulin, so insulin actually a protein. So when this insulin we are using, which is actually produced in different types of animal, this is foreign protein to us. So as a result of that, it can cause some immunogenic reaction, right? So, and it can vary person to person. So it is a problem. So nowadays, like I would say, currently we use all insulin, whatever insulin people use, all are recombinantly produced human insulin, not dog's insulin or pig's insulin, not like that. It's much before, long time back. Now, as you can see here, in 1980s, researchers used genetic engineering to manufacture a human insulin. This is the first time they use human insulin. Why 1980? Why not long time back? Because when I will be discussing in much more details about genetic engineering, you will be knowing that the problem is genetic engineering means you need some kind of tools and technique where you can actually manipulate our genes or genomes. But those tools and techniques discovered in 1970s mostly. So as a result of that, genetic engineering was not possible much before. Okay, and in 1982, the Eli Lilly Corporation produced a human insulin that became the first approved genetically engineered pharmaceutical product, right? So for human use, so this is the first product. And at present, as I already told, that researchers could produce genetically engineered insulin in unlimited supply without depending on animals. So how? The answer is through genetic engineering approach. Even I would say in India, one company, pharmaceutical company, Biocon. So Biocon produces a lot of insulin in their, in their company itself. Now, key words in genetic engineering. I would like to explain few terms in order to, discuss mechanistically like how we carry out genetic engineering. The first one is gene of interest. So here, gene of interest means DNA segment that is to be inserted or deleted. So for example, if I want to, delete some genes from some organism, or I would like to add an extra copy of gene into tomato plant. So for example, I already discussed that Bt cotton, that means we put some, genes from Bacillus thuringiensis bacteria to cotton plant. So that is our gene of interest, that gene which produces this toxin. So this is gene of interest. And then the next one is vector. So vector means here DNA molecule such as plasmid. So plasmid is an example of vector. There could be other type of vector also. I will be mostly discussing about plasmid in our discussion. So plasmids are small circular DNA, like small circular double-stranded DNA. So this is plasmid double-stranded DNA. There should be helix also. I am not showing here. And this is extra chromosomal. That means this plasmid is present naturally in some bacteria. So this is present naturally in some bacteria, but they are not part of their chromosome. Bacteria, they have one circular chromosome, right? In addition to that, some bacteria, they might have this plasmid, and these plasmids are very useful for our genetic engineering strategy, and I'm going to discuss soon. Now the idea is this is used as a vehicle to carry gene of interest into another cell where it can be expressed. For example, this plasmid, we modify this plasmid, we are giving some additional genes here. This is our, this is our gene of interest. So it will make some kind of protein which we are interested on, right? Now this plasmid, we can send it to, we can transfer it to a bacteria. So this bacteria, this is bacterial chromosome, right? This is a bacteria, and this is bacterial DNA chromosome. Now we are transferring this plasmid here in this cell, and now this bacteria will make the product from this gene itself, right? So as a result of that, although this gene of interest was initially not present in this DNA, but when we transfer this plasmid here in this bacteria cell, new cell, host cell we say, now that particular gene will be expressed. Express means, I discussed at the beginning that expression means here from gene to RNA and RNA to proteins. So the product we will be getting from this bacteria. Now this is called vector because sometime in school days we read that a mosquito is also a vector, right? So why mosquito is called vector? Because mosquito is taking some germs, and because of the mosquito bite, someone else getting this germ, right? So all of you know that malaria is carried by mosquito. So mosquito is called vector. So similarly, this kind of small circular DNA, which is called plasmid in this case. So plasmids are also called as vector for genetic engineering. Now transformation. Transformation means the transfer of gene of interest into a host cell, maybe a bacteria, where it can be maintained as well as expressed. What I already explained, that this plasmid was used to transform this bacteria. So as a result of that, whatever this bacteria was present before, now you have some additional property. It has, it is transformed. Why? Because some extra copy of gene we have already provided in this bacteria. So that's why it is transformed, and the process is called transformation. And the last keyword here, clone. Clone means organisms carrying identical genes. That's all. This is called clone. If we say about organism, then two organisms, they are exactly similar by by their genetic element, like they are carrying exactly same genetic information, then those two are cloned to each other. Okay. Similarly, if we make this plasmid again and again many copies, those plasmids are exactly same. So those plasmids are also cloned to each other. So that means they are exactly same. That's all. So here I am going to discuss the basic idea of genetic engineering. During our next lecture, I will be discussing more technical details about those things. Thing is, for example, if I say I am interested on some proteins which are present in some bacteria. For example, nowadays many detergent powder which we use for cleaning our clothes. So those detergent powder, they also contain some enzyme. So for example, I would say cellulase, lipase, those are enzymes. That means they are proteins. And now those proteins, if you add in detergent, they will work better. So for example, if you have some stain from some green grass, so you are playing in some, you are playing cricket for example, and you skid for a catch, and then some green stain may be in your trousers. And now if cellulase is present in detergent, it will kind of clean that stain very nicely. So as a result of that, this kind of enzyme nowadays we use in detergent. And this enzyme we can engineer also to make it more stable. Sometimes when we clean our cloth, we use little bit of hot water, so that if you engineer those enzymes in such a way that those enzymes will be kind of thermostable, so they can tolerate little bit more heat. So all those things you can do in lab. So you can engineer those genes which are producing cellulase, lipase, all those things. And then you can transfer that into E. coli strain and you can make more and more product, right? So I'm giving you just one a very simple example. For example, this cellulase, this enzyme you have taken. So this is a DNA, portion of DNA. So now this portion, a gene that codes for enzyme called cellulase. So what you will do? So you just use some specific primer to amplify this gene. So as a result of that, I already discussed about the PCR technique. So you amplify this gene. So as a result of that, this gene fragment, you will have many more copies, many copies of this gene. And now this is after PCR reaction. Okay, this is primer. Now you use some kind of plasmid. This is plasmid. This is circular plasmid. But now what you have to do? Somehow you have to use some enzymes. That enzymes will cleave here. So it will make a cut. And then you have to put your gene of interest here. So as a result of that, the new gene of interest, you put it here. So this is, I would say, cellulase producing gene. Okay. So now this plasmid you can use to transform some bacteria. So now this is bacterial chromosome and this is plasmid, and this bacteria now will produce cellulase enzyme for you. Okay. So the thing is, here this plasmid, when we are sending into this host cell, host bacteria, this is bacteria, we can control the number of plasmid, because most of the plasmid we used for genetic engineering, they have some kind of resistance gene. For example, most of them, they carry some kind of antibiotic resistance gene. So as a result of that, after transformation, when you are culturing this bacteria, if you increase the concentration of that particular antibiotic, like the antibiotic resistant gene is already present in the plasmid. So as a result of that, the bacteria will try to survive. So as a result of that, that plasmid will be copied more and more. So plasmid replication will happen. And this plasmid replication is independent of chromosome replication. So as a result of that, one bacteria might contain a huge number of plasmid. And remember, all these plasmid also contain your gene of interest. So as a result of that, they will produce huge amount of cellulase for you. This is kind of overall idea how we make our protein of interest in bacterial system. So I just gave example how to use cellulase or how to produce cellulase or lipase, like some proteins for human use, for example. Sometimes in research lab, we also use this kind of system for a little bit different purpose. For example, if I am interested on some proteins which are present in MTB, right? MTB means Mycobacterium tuberculosis. It's a very, I would say, like it's a pathogen. So as a result of that, Mycobacterium tuberculosis, this strain of bacteria you cannot use in lab here and there. You should have proper facility, that is called, biosafety level, like very high biosafety level facility should be there. So as a result of that, if I am interested on that protein, then how I can grow Mycobacterium tuberculosis in my lab to get the protein? It is not possible if I don't have that facility. Rather, what I can do, I can clone a gene from this bacteria, whatever I am interested on that particular protein. So as a result of that, you can like take some Mycobacterial tuberculosis genome, just the DNA, and then continue PCR reaction depending on, you have to use gene specific primer, that particular genes you are interested. On the basis of that, you have to design your primer, then you amplify that. So as a result of that, you will get gene of interest, right, which is coming from MTB or Mycobacterium tuberculosis. Then you just amplify. That means nothing but PCR reaction. And now you have many copies of this gene, right? And these genes you have to again send it to plasmid, and that plasmid should be transferred into laboratory strain of E. coli. So as a result of that, now you can make that Mycobacterium tuberculosis protein in E. coli itself. So that's why this is called heterologous protein expression. That means you are making this protein in E. coli, which is non-pathogenic laboratory strain, which you can use almost in all lab. But you are actually purifying the protein from Mycobacterium, you are getting the protein actually originated from Mycobacterium tuberculosis, but without culturing like Mycobacterium tuberculosis itself. So very commonly we do this kind of work in lab. So all those things are, because of this genetic engineering approach. So here genetic engineering means in bacteria we are adding extra copy of gene which is producing some MTB protein. That's all. Now this will be little bit different when we talk about eukaryotic gene, eukaryotic protein particularly. That's why I intentionally I provided some example of MTB protein. But for example, sometime during cell adhesion process, I discuss about E-cadherin a lot, right? E-cadherin. So which form adherence junction in our body, E-cadherin. So this is a membrane protein present in our body, like mostly on all epithelial cells this protein is present. Now if I am interested to work on E-cadherin, then we have to clone this, because you cannot really, it's very difficult to get like pure protein in high amount from original tissues, because you can say that we will take this protein from some epithelial tissues, but from where you will get the epithelial tissues that much? And again you have to purify that. It is a very challenging job. Much before, I would say, the genetic engineering approach, scientists, researchers, they used to do that, but now that completely it has been changed. We are relying on genetic engineering approach. So what we have to do, we have to clone this E-cadherin gene in some plasmid, right? So this is our E-cadherin, this is plasmid, and this is E-cadherin gene. So here now this E-cadherin gene containing plasmid you can express in suitable vector, a suitable host cell, not the vector. So host means you can express in bacterial system. Sometimes some protein you will not be able to express in bacterial system. During that time you might need to rely on eukaryotic expression system. For example, we often use yeast cell or mammalian expression system we often use. But I started this discussion because here you have to do little bit different tricks, because from how you clone this thing. So for example, if you isolate DNA from some human cell, like you have some human cell sample like human cell. So now from this human cell, so you can get DNA. And now if you use this DNA as template in order to carry out PCR reaction using some primer, DNA, this is template, and then do PCR reaction to amplify E-cadherin genes, and then you transfer that in this plasmid. But there will be problem if we want to express this in bacterial system, because like we discussed during transcription lecture that eukaryotic genes, they have introns, like, so they have exons and introns, but introns should not be present in mature mRNA. So as a result of that, you have to remove introns from your construct, from your gene of interest, which you are like putting into your plasmid. So as a result of that, you cannot rely on this thing. So no, this is not possible in this way. So rather we use a little bit different technique. So what we do, we isolate RNA from human cell sample. For example, some cell line you can use for that also, human cell line. So you have RNA. So RNA, but like we are interested on E-cadherin. E-cadherin is a protein, like cell surface protein. So that means this E-cadherin has been produced from some mRNA, messenger RNA, and this messenger RNA is being transcribed from the DNA itself, but we are relying on RNA. And particularly now, the major RNA present in our body is rRNA, tRNA, and mRNA. Apart from that, many other small RNA are also present, but for the time being, I am ignoring that. So now, it will be great if we can purify this messenger RNA, because that will actually enrich our initial sample. So messenger RNA, how you can purify? Because mature mRNA in eukaryotic system, they should have poly A tail at the 3' end, right? Poly A tail. So as a result of that, whatever the total RNA we have isolated, so this total RNA, again we can purify through one more step using some oligo DT. That means in purification, like in the purification system, if you use some like beds which are attached with poly DT, that means multiple T residue, they are attached with this some beds, then your poly A tail containing mRNA will be attached there. So major idea is here, through this method you are purifying mRNA, messenger RNA. From this messenger RNA, you can transcribe, reverse transcription that is called. Then what you will get? You will get cDNA, and this cDNA you have to use for PCR reaction, and then you can put that in some plasmid. So now this is your E-cadherin genes present in plasmid, but this E-cadherin gene, they don't have any introns, they have just exons. Why? Because we have isolated mature mRNA, they don't have any introns, and now that mature mRNA was converted into cDNA, and from there we made the PCR. So this is again very important step in case of eukaryotic protein. When you are working with eukaryotic protein, better to start with RNA and then make cDNA, so that you can ignore this introns part. So this is just a little bit of discussion. In the next class, we will be discussing in more details about genetic engineering. So that's all. You can follow any of this textbook. Thank you very much.